Hi, my name is Maha Dewar, and I'm going to be presenting the introduction and part of the methods section for this paper. The sperm oocyte decision is strongly influenced by somatic signaling pathways, where the expression of genes from somatic tissue can repress or stimulate the FOG1 and FOG3 genes, which are necessary for sex determination. The FOG1 and FOG3 genes are sperm-fated genes, but sexually transformed germ cells in FOG1 and FOG3 can produce oocytes. FOG1 and FOG3 are important regulators for sex determination. As I mentioned previously, FOG1 and FOG3 are influenced by somatic signaling pathways, which makes these genes terminal regulators in sex determination. And there are a number of genes that act upstream on FOG1 and FOG3 to control expression. These genes, for example, are TRA1, which is a transcription factor, FBF1 and FBF2, which is an RNA binding protein, and both can act directly on the FOG1 and FOG3 to repress or induce the expression of these genes. Other genes can also act on FBF1, FBF2, and TRA1 to induce or repress the expression. This is figure 1a from the paper, which shows the genes that act upstream on FOG1 and FOG3. As you can see, TRA1 and FBF1 and FBF2 can actually inhibit the expression of FOG1 and FOG3, which leads to oocyte differentiation. And if the genes are stimulated, then it leads to sperm differentiation. And there are also other genes, such as HER1, TRA2, FEM1, FEM2, and FEM3, that can inhibit TRA1, which can influence the expression of FOG1 or FOG3. FOG1 and FOG3 also have different functions in the cell, which are influenced by gene dosage, and it depends on the context in the cell. In FBF1 and for example, in FBF1 and FBF2 mutant backgrounds, FOG1 can stimulate cell proliferation, but it can also, in the same background, it can also inhibit FOG3 proliferation. FOG1 and FOG3 protein expression have a complex relationship with each other, and they're difficult to separate on a genomic scale. And the reason that they're difficult to differentiate on the genomic scale is because FOG1 and FOG3 have similar target sites in the genome, which makes it difficult to differentiate the mRNA of the two genes. So the hypothesis for the paper was that FOG1 and FOG3 proteins work together to control the expression in sex determination. In the methods of this paper, the researchers used the MOS-SAI, which is MOS1 mediates single copy insertion, where a single copy of the transgene can become inserted into the DNA in C. elegans, and the transgene becomes expressed in the genome and the germline. And the transgene are usually stable, and it can become inserted at a specific region in the chromosome. And this figure I got from the website shows the specific target sites in the 5 chromosomes of C. elegans, where a gene can be inserted into. MOS-SCI works by microinjecting DNA to generate an array. So you select the gene of interest, and with a MOS transpose, it gets removed, and this gene of interest becomes inserted into the chromosome, and it's expressed. To test the expression of whether the gene became inserted into the chromosome, you do a PCR analysis, and it will show the expression on the gel as shown in part C of this image. And you can also do a Southern blot to show the percentage of the genes that were inserted. And a sequence analysis also allows you to determine the gene expression. And here are the references.